Welcome to the lecture on bioenergetics. This is going to be a survey lecture where we talk about the exchange of energy and biological systems. So it'll have a flavor of thermodynamics, but also a flavor of reaction mechanics. So it turns out that as living beings, thermodynamics is going to be important for us. And we need to understand thermodynamic principles and ideas to understand how our cells and molecules react and how they change. And that can sometimes be very helpful for understanding disease, because disease looks at perturbations of reactions or structure or function. So some basic thermodynamics can be pretty helpful. Again, the lecture objectives are going to be really important for a lecture like this because there's a lot of detail here, but the lecture objectives will focus the study on the principles that are really important at the level of detail needed to make sense of this. So use the lecture objectives as a study tool. So we're going to be concerned primarily with free energy. And if you remember, free energy is the Gibbs free energy, or G, and a change in free energy is going to be a delta G. And we're going to focus primarily about whether a system, when it does a reaction, does it give off energy? Does it lose energy? Is that going to be a change in delta G where energy is liberated? Or do you have to put energy into the system to make a reaction go? Do you have to add energy, increase energy along those lines? So we're going to make a big deal about that. So in order to understand free energy and the change in free energy, it's helpful to look at the components of free energy. And the first one is the enthalpy, or H. And enthalpy is a function that's related to heat transfer, heat flow, heat movement. It also has to do with work expenditure in the system. So when you look at a delta H, or a change in enthalpy, you're asking what is the difference in the heat flow from state 1 to state 2. And by definition, the change in enthalpy is going to be the final state, whatever its enthalpy is, minus the initial state, whatever its enthalpy is, and that difference is going to be delta H. Now, delta H is going to report heat flow, and so if we have a negative delta H, that implies that heat is lost by the system, and that's going to be favorable. Or a positive delta H implies that heat has been gained by the system. So if you think about it, you have a cup of coffee and it's really hot and you set it on the desk. What's going to happen? The hot coffee is going to cool. It's going to lose heat to its environment. It will spontaneously cool down, and that's the spontaneous or the favorable process. If you took a mug of coffee at room temperature, it would not spontaneously heat up all by itself. You would have to pump energy into the system to increase the heat going in to increase the enthalpy. So it is unfavorable when you have a positive delta H. It is favorable when you have a negative delta H. And where this enthalpy comes in is like protein folding and unfolding all the weak interactions, binding energy, those kinds of things we talked about before. That's the main enthalpy component that you're going to see. The next thing we look at is entropy, or S, or a change in entropy is delta S. And a good fingertip definition is entropy is telling you a measure of disorder. Now, if you've had a lot of chemistry and thermodynamics, you know that there's a more correct definition of what entropy is, and it has to do with the availability or the unavailability of a system's energy to be used for work. And we know that nature favors a disordered system. It does not favor an ordered system. If you put something in, put a bunch of rocks in a line, perfectly in a line, you go away and you come back a year later, where are those rocks going to be? They're going to be all over the place. They're going to move over time. Things don't order themselves, they disorder themselves. So an increase in entropy is always favored. That's enough for entropy. So when we look at delta S, it's the same thing. If you're looking at a change in entropy, you're going to look at a change of state 1 to state 2. Delta S is going to be whatever the entropy is for state 2 minus whatever the entropy is for state 1. And if this number ends up being positive, that is going to be favorable. That means entropy has increased. If the number is negative, that is unfavorable. So you take delta H and you take temperature times delta S, and that gives you the free energy, and those two things go together. Now, that is enough of that. We're going to be concerned with free energy, and we're just going to talk about free energy just as a concept by itself. and that's here. It really is a hypothetical quantity. It tells us how much energy is available for work. And based on that, it allows you to assess whether or not any reaction that's going to take place will be spontaneous. And spontaneity really is a measure of probability. If you say something is spontaneous, that means it's likely to happen. If you say it's non-spontaneous, it means it's unlikely to happen. Not that it won't happen, but just unlikely. So when we look at the delta G is equal to delta H minus T delta S, okay, that you can see if delta G is negative, you can see how these two variables will work together to bring about that negative delta G. Let's just focus on delta G. Let's just look at that, okay? So again, if you're looking at a change in free energy, you're looking at these two states, and it's going to be state 2 minus state 1, and if it is negative, that is going to be favorable. If it's positive, it's going to be unfavorable. And remember, favorable simply means it's going to spontaneously happen, it'll happen by itself, it'll go. So how does free energy affect reactions? We're talking about biochemistry, we're talking about molecules reacting, we've talked about enzymes before and all this. We're looking at two substrates, A plus B, and there's going to be some chemical reaction where they have something going on and they come off as products, C plus D. And there's going to be some free energy associated with the change in state from state 2, state 1 here. Now for this reaction, if delta G is negative, that's what that says there, then you know it's going to be favorable for A plus B to go towards C plus D. You're going to expect to see more C plus D, you're going to expect to see less A plus B, because negative delta G says this reaction will proceed as written. Now, if you have a positive delta G for this reaction, positive delta G says it is not spontaneous. It's not likely to happen as written. That means you're going to have a lot of A plus B. You're not going to have much C plus D. That's what a positive delta G is telling you. Now, you don't know yet how much A and B you have or how much C and D. All you know is negative delta G says you're going to move to the right towards C and D, and a positive delta G says you're going to lie to the left, which is A plus B. That's what we're looking at right now. Now you may recognize there's a delta G, and you may have seen a delta G not with a little prime symbol there. And what's the difference there? Well, the difference is delta G is a really nice chemical term, but it's not very good for biological systems. It's at high pressure and high temperature and everything at 1 molar concentration. That's not what we have in our bodies. So the delta G not prime is telling us what is the delta G under biological conditions, 1 atmosphere of pressure, body temperature, and more normal concentrations. And in order to do that, in order to say how do these two relate to each other, we have an expression right here where delta G, the real delta G, is equal to that adjusted delta G plus this factor, which is going to make the difference between the two. And what is that factor? That factor is the equilibrium constant. Okay, wait. Let's back up a little bit. So let's look at the reaction A plus B, C plus D. And there's going to be a real delta G for that, where everything's at 1 molar and high pressure and high temperature, and you're going to get some equilibrium. But if you change the temperature and the concentrations and stuff, that's going to change the ratio. That's going to change the equilibrium a little bit, so you need a factor that will allow these two things to be related, and that's exactly what happens here. The standard free energy change under biological conditions is going to be related to the real delta G, but it's also going to have this RT log of KEQ in it. Okay, so what does this really mean? So for a reaction A plus B goes to C plus D, the equilibrium constant is going to be what's on the right divided by what's on the left. So the concentration of C times the concentration of D divided by the concentration of A times the concentration of B. That's the equilibrium constant. And if we set the real delta G equal to 0, you can do that. You can make whatever number up you want. We put the 0 right there, and we say, okay, if it's 0, what about this equation now? We could actually move the standard free energy symbol on one side, and we can put the RT log K on the other. So as you see this, what we're doing is we're actually moving the RT log K, and that's why it has a negative sign on it now. And so we can figure out what that biological free energy is based on the equilibrium constant here. R is the universal gas constant. T is the temperature of the system. Now, if you remember when we did this before, you have an equilibrium constant. And what you're looking at is you're looking at concentration of C and the concentration of D divided by the concentration of A and the concentration of B. And what matters is who's big and who's small. If you have a lot of C and D, these are going to be bigger numbers, and A and B are going to be smaller numbers. If you have a lot of C and D, it means you have low A and B. Under those conditions, C and D are big numbers, A and B are small numbers, and if you take a big number and divide it by a small number, what do you get? A really big number, and it's a big number here. So if the KEQ is greater than 1, which is what's going to happen, then the log of KEQ right here, the log of a number that's really big, is going to be a positive number. It's going to be positive. And then R and T are constants, and that negative sign is still sitting there. And so that means that your standard free energy is going to end up being negative. So when delta G, not prime, is negative, that means it favors C and D. Okay? Now what happens if you have a lot of A and B but only a little bit of C and D? If C and D are very small numbers and A and B are very big numbers, if you have a small number on top, a big number on the bottom, and you divide them, you're going to have a pretty small number. It's going to be very much smaller than 1. So in this case, if your KEQ is less than 1, if you take the log of a number that's less than 1, the answer ends up being negative. And so if this argument here becomes negative, you still got the R and T, but this negative sign over here will cancel, and you're left with a positive number for delta G, standard free energy here. And so what that means is when your standard free energy is positive, it means you're going to have a lot more A and B and not much C and D. You're going to lie to the left here. And then here's that rule of thumb just to remember again. We've talked about log base 10, now we're talking about natural logs, but the natural log is just 2.3 times the log of 10. So what we learned for the log of 10 is still true for the natural log as well. Okay, so let's take this one step farther. A plus B goes to C plus D. Here's your equilibrium constant. There's the C and D. What's on the right is on top. A and B on the left is on the bottom. And this is our relationship. And so we will say that if the delta G, standard free energy, is less than 0, log of KEQ is greater than 1, so C and D are favored. If your standard free energy is positive, it means the log of the equilibrium constant is a number lower than 1, and so your A and B are going to be favored under those conditions. And if you can remember that, you're all set. Negative delta G means you lie to the right. Positive delta G means you lie to the left. All right, so what's interesting about our bodies are we have all these reactions that are going on, and some are negative delta G and some are positive delta G. And how do we make sense of all that? Well, our body likes to couple reactions together. We know that negative delta G reactions are spontaneous. They will go forward as written. We know that positive delta G reactions are not spontaneous. They won't go forward as written. So how do we get those positive delta G reactions to take place? Well, we do a thing called coupling. We can take if there's a given reaction that has a positive delta G, let's say for A goes to B, and B goes to C. Let's say A to B has a positive delta G, 1.5 here, okay? So that means for 1.5 kilocalories per mole, whatever the units are, positive means it's going to lie to the left. You're going to have a lot more A, and you're not going to have much B. Now you have an equilibrium, so there will be some B, but there'll be more A. So how in the world can we get B to form under these conditions? Well, we can take that molecule B and we can react it to go to molecule C. And if that happens to be negative delta G, okay, if B to C has a negative delta G, then it means any B that's present will spontaneously form C. So B will go forward to C. All right, well, what happens then? When the B disappears, when B becomes C, the concentration of B is getting smaller and smaller and smaller. But remember, A and B is an equilibrium, and the ratio of those two must equal the equilibrium constant. So when you remove B, then some A automatically has to become B in order to keep that ratio correct. And so Between the A and the B, this B becomes a C. That means you pull more A over to B and B becomes C. You pull some A over to B and B becomes C. You can draw it over. And mathematically, you say you can add the delta G values for A to B and B to C, and you can get the overall delta G for A to C. So if A going to B has a positive 1.5 kilocalories per mole, and B to C has negative 3.5 kilocalories per mole, when you add those things up together, you're left with negative 2 kilocalories per mole. So that tells you A going to C overall is going to be favorable or spontaneous. So let's see an example about this. We've already talked about the hexokinase, glucokinase enzymes a little bit. That's where ATP and glucose come together in the enzyme active site. And the phosphate from ATP is transferred onto glucose to make glucose 6-phosphate, and the triphosphate ATP becomes the diphosphate ADP. And the overall delta G for this reaction is negative 21 kilojoules per mole. Okay, so because that is negative, that tells you that this process is spontaneous. This reaction will go forward. Why is that? Well, if you have glucose and you just have an inorganic phosphate, if you want to stick those two together with a new covalent bond, you have to pump in 14 kilojoules per mole. Okay, that's positive. That's not spontaneous. That's something you're going to have to add energy in. But if you couple that with removing a phosphate from ADP, ATP to ADP plus phosphate, that's negative 35 kilojoules per mole. So when the ATP is split, there's enough negative delta G that can more than make up for the positive delta G, and the net ends up being negative 21 kilojoules per mole. So it's a favorable reaction that goes forward. This is going to happen in a lot of the pathways that we see, and the TCA cycle is really a good example. Again, we'll have a lecture on the TCA cycle coming up pretty soon, but you can look at the numbers here and get an idea. You don't have to memorize this. If there's any kind of question, I would show you this and I would ask you about it. When you look at the TCA cycle, you'll see when you look at the delta G values for the different reactions, some of them are negative, some of them are positive, and a couple are 0. Wow, okay. So the negative delta G is telling you that this is a spontaneous reaction. It will go forward as written. Acetyl-CoA will mix with oxaloacetate to produce citrate and coenzyme A. That's going to happen. But this positive delta G right here says, wow, the reactant, the left side is going to be favored. Citrate is favored, and you're going to have less isocitrate formed. In fact, that's what those arrows are showing you right there. You see the arrow lies heavily to the left and just a little bit to the right, and that's because of that positive delta G. Up here, you see that strong dark arrow saying you're lying hard to the right here, and that's because of the negative delta G right there. Okay, well, does the TCA cycle stop? Is it done because you've got this positive delta G? Well, not at all. Look what comes next. Isocitrate to alpha-ketoglutarate has negative 5.3 kilocalories per mole. And because of that, whenever an isocitrate shows up, that isocitrate will spontaneously move down to alpha-ketoglutarate. And whenever you consume isocitrate, you reduce the concentration of this molecule. That messes up this equilibrium right here. And so some citrate will be automatically converted to isocitrate to reestablish the concentration and the ratio of those two molecules. So you can actually pull the reaction through this positive delta G by drawing it around to alpha-ketoglutarate. And of course, once you get to alpha-ketoglutarate, zoom, you get into succinyl-CoA, and then succinyl-CoA will move to succinate. You have a little bit of a negative delta G here, but enough to keep things moving. This 0 kilocalories per mole simply says none of these are favored over the others. You're going to have a pool of malate, a pool of fumarate, a pool of succinate, which means an amount or a collection of them. And these molecules can go back and forth fairly easily here. And now what do you have? You have a positive delta G, a very large positive, plus 7 kilocalories. You don't have to memorize these numbers. You don't. But understand the idea. And you see this has a very dark arrow laying to the left and a very thin arrow lying to the right. So that means that malate is going to be favored under most conditions. But again, because of this negative delta G here, which is slightly more negative than the positive 7.1, oxaloacetate will be drawn over to citrate, and that will reduce the oxaloacetate concentration, and so malate will convert to oxaloacetate to reestablish that equilibrium, and you can move through there. This turns out to be so incredibly important for the TCA cycle and for energy metabolism in our cell, because we're going to find out that these enzymes are allosterically regulated. The ones that have large negative delta Gs are going to have allosteric modulators that will turn them on or turn them off based on what the energy needs are for the cell. And that will affect whether or not the enzyme will do the reaction or not, which is how you control these large negative delta G reactions. Okay, so let's bring in the idea of a high energy biomolecule. And the idea that we had before was if you have a positive delta G reaction, but you need that reaction to take place, it's a good idea to couple it together with a molecule that has a lot of free energy that will have a negative delta G when it reacts. And high energy biomolecules are exactly those kinds of molecules. So you can use these high energy biomolecules and you can let them react so they liberate energy, and that energy can be captured by the enzyme to drive the positive delta G. And we're going to see lots and lots of examples. We're going to see high energy phosphates, and we're going to see electron carriers like we've seen in the DH and FADH2 so far. So we'll see some examples here. And that brings up the whole idea of energy transfer. You want to be able to move energy from one place in the cell to the other, either to drive a reaction or to allow work or to allow transport or to allow something to happen here. And in order to do that, we know that there is energy contained in electrons. And so these coenzymes that carry electrons like NADH, the hydride, or the FADH2, 2 electrons, 2 protons, they can carry that energy. And the high energy phosphates are going to carry energy because that phosphoanhydride bond, which we'll see in a moment, is a very energetic bond. And so just the fact of having a phosphate oxygen phosphate will give a lot of energy. So here, let's look at the high energy phosphate compounds, and that's going to be for our energy transfer. So we have a molecule like UTP here. UTP is similar to ATP, adenosine versus uridine. But you have 3 phosphates. And so each of these phosphate oxygen phosphate is a phosphoanhydride bond. And that has a lot of strain. There's a lot of negative charge and a lot of electron density. And so there's a lot of strain in this molecule, which makes it it has a lot of energy contained within these bonds. So a triphosphate is considered a high energy phosphate compound. Over here, you've got a single phosphate on this glucose molecule. In fact, it's a carbon oxygen phosphate. So that's called a mixed anhydride. We have carbon oxygen phosphorus. It's got some energy because of that phosphate, but not nearly as much energy as the UTP molecule has. Well, glucose 1-phosphate has some energy, but it doesn't have quite enough energy to do what it needs to do in the cell. We need to find a way to increase the energy of this molecule. And we can do that by taking the energy that's contained within this phosphoanhydride linkage here, this phosphate, a pyrophosphate molecule right here. If we could somehow create a dual phosphate on this molecule, we would raise the energy status. And you can see you can do that. There is an enzyme that will take these together, and you connect the phosphate onto the UTP molecule. You eliminate that pyrophosphate right there. That's gone now. There's the pyrophosphate that's the product. But this 1-phosphate that's attached to the uridine now is connected to the phosphate that was on the sugar. And you have created a phosphate oxygen phosphate, that phosphoanhydride linkage. And so now this glucose molecule has every bit as much energy as a diphosphate molecule like an ADP or a UDP or whatever. You greatly increase the energy status of this molecule. And that's because you took the energy of the phosphate and you incorporated it into that molecule. Okay, so UTP has a lot of energy, UDP glucose has a lot of energy. What's going to prevent these two from just immediately back reacting and forming the UTP? What happens? Well, the cell's pretty smart. The cell says right now this pyrophosphate has an awful lot of energy and it can back react to give us the starting material. So the cell will say, I'm going to hydrolyze. I'm going to add water across this so I break that phosphoanhydride bond and I'll have two inorganic phosphates. And once you break that pyrophosphate up and you liberate that energy, there's no way you can get those two individual phosphates to come back together to make a pyrophosphate. It's just not going to happen. And so by hydrolyzing this pyrophosphate, two things happen. First, the energy that's liberated by hydrolyzing this pyrophosphate is the energy that is used to drive this reaction right here. That's the energy required to take the energy off of this triphosphate to create the diphosphate molecule here. And then the second thing that happens is once you make individual pyrophosphates, you've now made this thing basically irreversible. It can't go backwards. So a really smart concept of the cell, a really great way that this works. And where we see this is in glycogen synthesis. We've talked a little bit about how the liver or skeletal muscle will take free glucose and it will use that free glucose to make a very large polymer glucose, which is glycogen. And in this case, glucose comes into the cell. The glucose itself doesn't have a lot of energy, but the kinase is going to take ATP and it's going to use that to phosphorylate the glucose. So now glucose 6-phosphate has a little bit more energy than just plain glucose. It's also now trapped in the cell. And then you take that glucose 1-phosphate and you react it with UTP, just like we saw in the previous slide. You're going to knock off an inorganic phosphate, but in the process, you are going to create that UDP glucose molecule. And meanwhile, that pyrophosphate is going to be hydrolyzed, and you'll get two inorganic phosphates there. So in the process of the UDP glucose being formed is now very high energy, and you can take glycogen, and you can add that glucose residue onto the glycogen, and you liberate that UDP out there because it has energy. And we'll see how that works in the glycogen lecture. So here's the idea of transferring energy from ATP. You give that energy to the glucose, and then you take the energy of a UTP, you give that to the glucose phosphate, you create the very high energy UDP glucose, and that allows this reaction to take place. So it ends up being all relative. That's what matters in the cell. And there are some molecules that have a lot of free energy that they can use, and there's some molecules that have lower free energy. And that's okay. And it's just important to remember what's high and what's low. We will see, we haven't done glycolysis yet, but we will. There's a couple of molecules in glycolysis that are used to create ATP during that glycolytic pathway. That's the idea of the anaerobic ATP production. You can create ATP directly in the cytoplasm of the cell without using molecular oxygen. And that's because phosphoenolpyruvate and 1,3-bisphosphoglycerate have more free energy than ATP has. So they can transfer a phosphate from themselves onto ADP, and they can make ATP along the way. There's also a molecule called creatine phosphate in muscle, which we'll talk about pretty soon. And creatine phosphate also has a high energy, and it can be used to phosphorylate an ADP molecule to recreate ATP inside the muscle. So these are higher than ATP, so they're used to make ATP. But then ATP is going to be used to phosphorylate glucose or phosphorylate glycerol or phosphorylate other molecules, proteins, or whatever like that, because ATP has enough energy to work on these lower energy molecules here. So what's in the middle? ATP. Actually, all nucleotide triphosphates are in the middle. And ADP isn't bad either. ADP has about the same energy as ATP because ADP also has that phosphoanhydride bond in it. And then pyrophosphate we saw before, those are those two inorganic phosphates that are linked together. It's got the same energy as a phosphoanhydride bond in the ATP or ADP as well. But then down here we're looking at glucose phosphate, we're looking at glycerol phosphate. These are organic molecules that just have a single phosphate attached to a carbon. They've got more energy than, say, glucose or glycerol by itself, but it's not as high as like ATP, for example. And that brings up the concept of ATP being the energy currency of the cell. We can create ATP by oxidative phosphorylation. We can create ATP by anaerobic ATP production with glycolysis. It's a molecule that can be made, and then once that molecule is made, it moves all around the cell, and it can be used to do the work of the cell. It can give energy to molecules, phosphorylate things, can participate in reactions or whatever. It's easily made, it's readily spent, and it's crucial for all of our metabolism. In fact, it is pretty well understood now that we pretty much make and use our body weight of ATP every day in all of our cells. That's how much ATP is being used on a daily basis. It's kind of a big deal. Okay, so high energy phosphate molecules are one type of molecule that carry energy, and the other is going to be these electron shuttles. Electrons have energy, but electrons can't exist just by themselves in solution. They need to have a home. And so these cofactor molecules, here is the flavin, in this case FAD is present here, can accept 2 electrons and 2 protons, and they can go to form these resonance structures of this ring system. Remember that from the vitamins lecture and the cofactors lecture. The other one over here is the nicotinamide adenine dinucleotide molecule here, where the hydride, it's a proton with 2 electrons, and that's an entire covalent bond, and it can be attached to this carbon, and that'll carry those electrons safely as a covalent bond. So these are molecules that are oxidized, and then when they receive the electrons, they become reduced, and then they pass the electrons off to somebody else, they become oxidized again. So but the term shuttle just simply means you are receiving electrons, NADH will move them around the cell, FADH2 will just hand them off to some other protein, and we'll see how they work coming up pretty soon. So here's the overall idea of energy transfer in the mitochondria with electrons. We've talked about this before, that the TCA cycle exists to pull electrons out of molecules. That's what the TCA cycle is. It's just oxidizing, oxidizing, oxidizing. It's pulling those electrons out, and it's giving those electrons to NAD to form NADH. It's giving those electrons to FAD to form FADH2. And those are going to be the electron shuttle molecules. Then those molecules are going to carry the electrons over here to the electron transport chain, and they hand those electrons off. And so you've transferred the energy from the molecules of the TCA cycle, and that energy has now been given to the electron transport chain. Well, the electrons will move through the electron transport chain to set up a proton gradient, which we'll talk about when we do the electron transport chain. And that proton gradient is going to be used as an energy source to drive ATP synthesis. So the energy that's obtained from the electrons as they move through the electron transport chain is used to create a gradient. And if you remember from the membrane transport lecture, whenever you have an ion gradient, there is energy, potential energy held in that ion gradient. And the ATP synthase over here will take advantage of the proton gradient and the energy liberated by letting the protons move through the membrane, and that's the energy that's going to be used to help synthesize the ATP, as shown here. So there's actually a series of redox cofactors, of course, NAD, NADH, FAD, FADH2, or FMN. Those are very similar. So the nicotinamide, here's the flavins. Coenzyme Q is a molecule that's in the intermitochondrial membrane that's going to shuttle electrons around. Lipoic acid, it's a disulfide molecule. We'll see that when we do the TCA cycle. Coenzyme Q we'll see when we do the electron transport chain. Vitamin C we're going to see when we do the free radical lecture and toxic oxygen intermediates. And tetrahydrobiopterin we'll see towards the end of the class. And that's going to be similar to tetrahydrofolate as well. These are cofactors that can carry electrons. Okay, so what do we talk about when we say electrons have energy and they do energy transduction? So this is kind of important for the MCAT when you think about the standard reduction potential. And the standard reduction potential is going to tell you how likely it is that an electron can move from one molecule to another. So just like we've seen for delta H or delta G or delta S, there's going to be a favorable movement of electron based on what the potential is for these two different molecules. Now, what we find is that the reduced coenzymes, when they have these electrons, they have to receive electrons. So whatever process is used to extract electrons and put them on the oxidized NAD to make NADH, or the oxidized FAD to make FADH2, that has to be a favorable movement. And then when these reduced coenzymes get to the electron transport chain, when they dump their electrons off, the movement of the electrons off of these cofactors onto the chain also has to be a favorable movement. And that's going to allow the electrons to move from the TCA cycle onto the cofactors, from the cofactors to the electron transport chain, and then through the electron transport chain to help set up that gradient. How's that going to work? So we look at this basically as the redox, something that is reduced, something that is oxidized. If it's reduced, it has the electrons. If it's oxidized, it does not have the electrons. And during the reaction, what is reduced can give an electron away. It's going to become oxidized. During the reaction, something that is oxidized can receive the electron. It's going to become reduced. So that is the oxidation reduction reaction going on. So you can break these things into two half reactions where you say a reduced molecule is going to become an oxidized molecule plus an electron, or an oxidized molecule plus an electron can become a reduced molecule. And that's typically what the MCAT will work with, these kind of things here. But the two half reactions go on at the same time. So that brings us to the standard reduction potential, and that tells you how likely is it that an electron will move from one molecule to another. And we know that electrons will spontaneously flow from a molecule that has a more negative standard reduction potential to a molecule with a more positive standard reduction potential. And thank goodness that makes sense. That's how I remember it. I'm thinking if I'm a very negative molecule and I'm beside a more positive molecule, then my electron is going to be attracted to that more positive molecule and it's going to spontaneously move to the more positive molecule. That's how I remember this here. Okay, so we can look at how this works just by looking at some standard reduction potentials. And this, actually, we'll look at that in just a minute. I'm sorry, I got ahead of myself. So here's the energy transfer in the cell where you can use high energy phosphate molecules. You can phosphorylate proteins and that changes their activity. You can hydrolyze ATP for energy, like for muscle contraction. And when the ATP level gets kind of low, you can phosphorylate ATP to make ATP. These are all the examples that we've seen. Of course, we have the reduced cofactors going to the electron transport chain to help create the proton gradient that's going to help create ATP synthesis. Again, lots and lots and lots of lectures coming up to cover each one of these systems here. So when we put it all together, we look at what's going on in the mitochondria. And you think about all the oxygen that we consume on a given day or whatever. You breathe in oxygen, that oxygen is being used by the body. The vast majority of the oxygen that we breathe every day is going to be used by the mitochondria for oxidative phosphorylation. That means oxygen-dependent synthesis of ATP. That's how serious our oxidative phosphorylation pathways are. There's going to be a little bit of oxygen used for other things like reactions where you have to get oxygen atoms and stuff. And we'll see a few of those coming up here. Okay, so if you think about the mitochondria and all the oxygen it uses, what is that oxygen being used for? Again, the vast majority of that oxygen is used to create that ATP. But there's a thing called the proton leak, which is it's called an uncoupler. And we'll see that when we do the electron transport chain. Little bits of protons can slip through the membrane. Instead of making ATP, you can dissipate that energy as heat. And that's one of the heat responses that we have to help us warm up. Now, if you're thinking about ATP synthesis, what is that ATP going to be used for in our cells? Well, it's going to be used to synthesize protein, establish ion gradients. It's going to help with calcium and muscle contraction. It's going to be necessary for synthesizing glucose, synthesizing urea, all of our muscle contraction processes. There's so many things that require ATP energy to make them go, which is why we have so much ATP synthesis, which is why we need so much oxygen on a regular basis. Okay, so this is a really good example of a test question here. Let's consider the reaction A plus B, and that's in equilibrium to C and D, which are the products. And it says it's at equilibrium, so you know you can apply those rules we're talking about. If the equilibrium constant, KEQ, is 1 times 10 to the minus 3, what can be said about the delta G, not 0 value for the reaction as written? Okay, so the first thing you ask yourself is you're saying, well, is this going to have a positive delta G or is it going to have a negative delta G, and how do I know? Well, you know, if it's a positive delta G, it's going to lie to the left. A negative delta G, it's going to lie to the right. Well, that's what they're asking me. They want to know which one is true. Well, the KEQ is the next thing you talk about. If you remember, when you have an equilibrium constant, If C and D is big and A and B are small, that equilibrium constant is going to be a number greater than 1. But if the equilibrium constant is a number less than 1, it means that A and B are big and C and D are little, because C and D are on the top, little number, divided by A and B on the bottom, big number. Okay, so if that's the case and KEQ is less than 1, that would suggest that A and B are going to be bigger numbers. And so that suggests that this equilibrium lies to the left, and if it lies to the left, then it must be a negative delta G. So hopefully you saw negative delta G and you picked that as your answer. So that's where you come from here. Because C and D are small, A and B are big, that ends up giving you a positive delta G value, and that is your answer. Okay, here you're studying a reaction. Again, A goes to B and B goes to C. And we're telling you that the delta G not prime value is going to be for A and B is positive, so it's positive 10. And the delta G value for B going to C is negative 15 kilocalories per mole. Okay, so which of the following best predicts how this reaction will proceed? Now it's kind of a hard thing because you don't have an immediate answer. You have to look at the answers to figure out which one is going on. You have to think through the answer choices. But if you answer this first and then find your right answer, you're going to be confident that you have answered the question properly. So what do you know? If A and B is positive 10, and you know B and C is negative 15, you can combine those together. And positive 10 plus negative 15 adds up to negative 5. And if the overall reaction has a negative delta G, it means it's going to lie towards C. So what you're going to look for is some answer choice that has something to do with C being the biggest. So let's see if we have that. A will be present in the greatest amount, B and C, oh there it is, C will be present in the greatest amount. And that's what you're looking for. And you've already reasoned through your answer. You know that's the right answer. So you have that double confirmation that that's going to be correct. And that's where the answer lies down here. You just add those delta G values up. They're negative. It means you lie to the right. And there it is. Kinase enzymes usually catalyze the transfer of phosphate from ATP to a target molecule. All the time. You always see kinases like protein kinase A will bind ATP and it puts a phosphate on a protein. Or hexokinase takes an ATP molecule and it sticks a phosphate on glucose. That's what kinases typically do. However, there is an enzyme called pyruvate kinase that actually catalyzes the creation of ATP using the molecule phosphoenolpyruvate, and that reaction has a negative delta G value. How is this thermodynamically possible? Well, wait 1 minute. Okay, wait. So ATP has energy, negative delta G, it's a pretty strong molecule. But you recall there's a couple of molecules that have a greater negative delta G when they react. And phosphoenolpyruvate is one of those molecules that has more free energy than ATP. So phosphoenolpyruvate can take one of its phosphates and it can stick it on an ADP to create ATP and still have a negative delta G. And that's what's going on here with the pyruvate kinase enzyme. If you remember a couple of slides ago, there's 1,3-bisphosphoglycerate and there is phosphoenolpyruvate. Those are two molecules that are used to make ATP in the glycolytic pathway. We'll see those in the glycolysis lecture coming up in just a couple more lectures. So what you're looking for is you're looking for an answer that talks about phosphoenolpyruvate as having more energy than an ATP molecule. And do you see it down there somewhere? There's more PEP than ATP quickly. PEP has more free energy than ATP, so it can phosphorylate ADP. That's your answer right there. And again, because you thought through it and you found out what the answer was, it was easy to find the correct answer here in the possibilities. And that is what you have. Here you're studying an oxidation reduction reaction. A goes to B. Species A has a standard reduction potential of negative 10 millivolts. Species B has a standard reduction potential of 5 millivolts, positive 5. For this reaction, which way will the electron spontaneously move? Okay, you're wondering where the electron is going. And if I recall right, standard reduction potential says if you're more negative, the electron will move spontaneously to something that is more positive. And so here A is negative 10 and B is positive 5, which is, so A is more negative, B is more positive. So I'm going to say that it goes from A to B. And that's exactly what's happening, right? It's going from more negative to more positive. So which answer is that? It is going answer A, from A to B. Yeah, that's the one right there.